Okay, thank you everyone for coming. It's my great pleasure to introduce Judy Jinn as her exit talk, which is not really, it's one of these, our pseudo exit talks this semester, because Judy is actually coming to Harvard next year. I'm going to be at Harvard on sabbatical, and Judy has a year of her NSF, so she's finishing her data collection, and then she's going to leisurely write her thesis in David's lab at Harvard next fall and spring if she needs it, but she may be done by Christmas. So this is her Tolman exit. Judy came to me from University of Michigan, where she got a double degree in biology and psychology in 2012, and along with being a nationally ranked Taekwondo black belt and a graphic designer and recently about to go to the Nationals on color and dance. Those are her little sidelines. Judy trained 20 species of wasps to recognize faces as an undergraduate, which resulted in, it was part of a big group by the group in Michigan that went to became science paper, the first insect to individually to recognize individuals by face. It was really an amazing study. But Judy was the undergraduate who was training 20 species to make sure that it was just this one, these few specialized species that could do it. Then, for the summer, she went and studied dolphins and trained and studied imitation dolphins. So went from the wasp to the dolphin, and got to Berkeley. In her first year, led the study to show that gecko, the lizard, can walk on water. Not just Judy, the geckos too. And in fact, in that paper, she's the lead author. Is also now about to be submitted. And then she moved to squirrels. Those are our lab obsession squirrels. And then she decided to move into navigation, and that would be the topic of today's talk. She, in the last few years, we basically invented the study of looking at olfactory navigation in humans and in dogs, and she's been concurrently running 4 experiments, some of which she'll talk to you about today. It's a great pleasure to introduce you.
Alicia, that made me sound a lot more awesome than I really am. Thanks. Thank you all for coming. I realized last night when I sent out that email and Facebook invite that I actually didn't tell half of you what I actually do for my research. So if you came for a squirrel talk, I apologize, but we can talk about it later. We're talking about human and dog behavior during olfaction. So first of all, why would you even care about olfaction, right? So first of all, it's one of the most understudied modalities because as humans, we rely on vision, right? But actually, if you look at the physiology of vertebrates here, read my mouth if people are watching this talk online, that purple line here, that delineates the first appearance of olfactory receptor genes. And that was over 700 million years ago. It appeared as tiny fish-like creature, which has a very basal spinal cord. And you can see it's been conserved all through the lineages of animal taxa that we know today. And it actually diverged into the taste system that we have as well. So those two things are very tightly linked in our brain. The thing is, though, that even though it's been conserved a lot, humans don't really use it, so we've ignored it, right? And it wasn't until 2004 that there's a Nobel Prize awarded to Richard Axel and Linda Buck for actually understanding what odor receptors do, how they're organized. And despite that Nobel Prize being awarded for a 1991 study, I think, we still don't really know what happens between when the odor hits our nose and then it gets to our brain and then it's actually perceived, right? You can think of, as humans, we have 400 unique odor receptor cells, but we can identify 10,000 odors, which is pretty good for a primate, which doesn't really use smell for much besides cooking, right? The thing is, though, we can think of it as an alphabet. So the letters are the odor receptors, and the words are what are formed when certain neurons are activated. But what happens in that black box is unclear. But if you look at brains, this is a rat brain right here. And the first thing you're going to notice is this giant structure out front. That is an olfactory bulb in a rat. So animals are very reliant on this, some more than others. But in all vertebrates, what happens is after the animal smells something, triggers the neurons, right? Inside the olfactory bulb, they perceive a scent. It is then projected over to the entorhinal cortex, where we find grid cells. And from the entorhinal cortex, it attaches to the hippocampus. We know the hippocampus, the psychologist here, very important for spatial navigation and memory. Also the place of where place cells are located. And so grid cells and place cells, for people who don't know, was a 2014 Nobel Peace Prize, I wish, the Nobel Prize, right? That was the Mosers and John O'Keefe. And so this really like conserved circuit in all vertebrates, there's something important about how smells go from the nose and then get to our spatial navigation. But nobody really has studied that intensively, right? And not only this, but the olfactory bulb and the hippocampus, those are the only parts of our brain that have adult neurogenesis. And that means new neurons are made in these parts of the brain, not like they're just sitting and doing nothing. Right? So something's important here. 2 Nobel Prizes were awarded in the past 20 years for these parts of the brain, so we should really care about this. So like, what is my role in this, right? So I'm not a neuroscientist, I'm not an evolutionary biologist, that's not my background. I really care about behavior, and so I really want to study why we have olfactory navigation and what its use is. All right. So, olfactory navigation. The thing I want to ask all of you here is, if I were to actually picture in your head the quintessential animal that uses odor to get around, what would it be? Shout it out. I'm sure we're all thinking something. Dogs, exactly, right? So does anybody here actually know what is the most studied animal in terms of navigation, olfactory navigation? I'm gonna have a guess. A rat. That would be my first guess too, right? That is not true. I have looked extensively. There's actually not a lot of studies about rats navigating. There's a ton of studies showing like rats can like odor discriminate, right? They can like stick their nose in a hole if there's an odor coming out of it and like do a no-go situation. Actually, the animal that's been most studied for navigation, which is still not very much, is homing pigeons. They're birds. And so how many out there actually thought birds have no sense of smell? Yeah, I did not know that they had a sense of smell either for a long time. Homing pigeons actually are great sense of smell. So what I'm showing here, the figure on the left side, this is a control bird. When it flies home, it goes in this like northeasterly direction. For a long time, people thought that homing pigeons were using magnetic fields to get home, right? So the obvious study for psychology and neuroscience is like, let's just lesion that nerve, see if birds can still go home, right? That's the middle graph here. Birds are still going home. But this one on the far right, if you lesion parts of the brain for olfactory navigation and olfaction, you can see these birds, they just kind of like go in random directions. So it turns out homing pigeons use the smell to fly home. And the second most important animal that has been studied for olfactory navigation are actually invertebrates. So you can see down here, this is a scent cone. And the reason scent cones are important is because scent does not just like laminarly flow out in like a gradient. If you imagine like a plume of smoke, there's actually directionality to it, right? So you can actually exit a scent cone. And so what they're showing here is that invertebrates, they like to go in and out of the scent cone. We call this pattern casting. And in fact, if you look at birds and fish, other invertebrates like moths, salmon, they all develop this sort of casting behavior. And what they're doing is going in and out the edges of the scent plume, finding that they're not in it anymore, and then switching and going back again. And then they go up the odor gradient until they find the source. All right, so the most obviously missing animal from here are all the mammals. So all the psychologists in the room know we study rats and mice like all the time. Why has nobody done a single study about this? I think there's like maybe a paper about it, maybe one. So we have collaborators doing this right now, but this is like a horribly deficient area, right? This should be like what we're thinking of first. So I have some main questions I want to address in my dissertation. And one of those like hypotheses I have is that mammals should exhibit the exact same kind of odor tracking behavior, this casting behavior, as all the other invertebrates as well, because it's such a highly conserved gene in our system, right? And I have another question too, and I'm going to show you a bunch of pictures here of noses. And it's kind of a trick question, not really though, but looking at all these images, what is the common trend among all of them? There's 2, exactly, good job, yay! There are 2. The one tricky one here is bottlenose dolphin, it has 1 blowhole, so keep that in mind, I'm gonna come back to that. So the question becomes, why do we have a paired sensory structure for olfaction, right? Evolution does not conserve things based on if they're useless. Like cavefish, they're gonna lose their eyes because they don't use them anymore. But for some reason, through all these animals, we have 2. We have 2 nostrils, right? 2 antennae on these moths, even the fish have them too. So then I'm gonna hypothesize that having a pair of sensory structures actually improves olfactory navigation. And finally, I'm gonna show you this odor plume here. Note that it is extremely complex. So odors move through turbulent or laminar flows, usually turbulent though, right? We wanna know how animals are actually sampling an odor plume and then getting through it. So exactly what are they smelling and where do they go next? We have a big NSF grant with 7 PIs. They're studying odor plume navigation just like we are. John Kamali's lab in Boulder. He's the one who took this image, and he got this image by having a plane of lasers shooting through a plume. This is in a water flume. And the first thing you're gonna notice is if you've never had a fluid dynamics course in your life, this is a very complex structure. And there's actually a lot more empty space than you might realize. And this, if I was playing in motion, I'll show you a video later, it's not easy to predict. That's why it's been a huge question for a very long time. There are other people in this NSF group who are modeling animal navigation, and they're showing pretty well that, yeah, animals should be able to navigate a plume. We know in real life they do that too, right? And I think models are great. They have done fantastic work, but in order to prove a model or support it, you want to have empirical evidence, right? So ideally what you do is like you take your rat or your mouse and you chuck it into this laser, and then you see, where does the rat or the mouse go? What is odor plume? Can you like line these things up? Well, it turns out that laser is so strong, it can ignite dust particles in the air. So the last thing you want to do is chucking mice into this thing, right? So we thought, animals are so good at this, we don't get it, but clearly they're doing it. What if we use animals as kind of like an odor detector proxy? If we study where they're going and they know where the odor is, we study enough of those animals, we should be able to track kind of what happens to an odor plume when it has like the effect of wind, temperature, humidity, right? So that's another question I'm going to have, and I'm really going to focus on the fact that navigation should be more difficult in high winds, low humidity, and high temperatures. Okay, and to kind of break up this talk a little more, make it easier on you guys, so I'm always going to be talking number one, which is the casting behavior for all these studies, but then I'm also going to focus on stereolfaction and odor dispersion. So in terms of stereolfaction, we're going to look at that in humans. I have a virtual odor task and an indoor search experiment. And then odor dispersion, which you all came for, the K9 experiment. We're going to talk about the meteorological effects of how odor is dispersed. Okay, so here's that video I promised of a plume moving through in real time. The thing is, since we don't really know what mammals are doing, I want to really abstract this idea, and just all I care about was what is the concentration of odor at the time of the sample, the sniff, and what is the next step that the animal takes right afterwards. So in order to do this, I had my friend create a little game for me. So this is a computer game. People walk around just using the arrow keys. They have to hit the space bar in order to sample the environment. And what they get is an audio cue, which is directly proportional to how intense that odor is. So that odor right there that you're seeing walk around, that's another odor that was sampled by Kamali Lab in Boulder. So that's exactly what a plume does in real life. You can hear my computer, but the closer you get to the source, which is their goal, louder it gets. And people cannot see that plume. It's totally visible. So they're walking around, just seeing the space bar. When they think they're like right on top of it, they just hit enter, and that's their guess. And then I also further divided these people into three groups. So there's a monocondition group, a stereo group, and a flip stereo. So stereo just means if the plume's on your right side, it's going to be louder on that side, hopefully for you, right? Flip means I'm going to confuse people, flip the right and left inputs. And finally, we also gave everybody there a little survey for the questions by Haggerty. This is a survey that's been used for analyzing people's sense of direction. And it's really simple, like really useful actually. It's just questions like, I get lost very easily, I like reading maps, I can tell other people directions. I just kind of like take all the scores and I assign people a score 1 through 7, 7 being the highest for having a good sense of direction. So on the right side here, this is a video of a person actually playing the game. And every time the arrow flashes red, right there, I'll point on the computer, that is when they actually are hitting the spacebar. So they're sampling frequency. And you can see the dark gray there. That's when they're like starting to like, looks like a casting behavior. So the advantage of this computer game is that it removes a problem for me for actually tracking an odor in real life and using lasers or something to photograph odor plumes. I can track movements of the subjects extremely easily. And I'm recording the sampling frequency. And also this interface is totally modular. So I can move the plumes around, I can change the size, the sampling space, I can change the shape of, or like make it into like antenna type things, a whole bunch of things. It's really nice. So each person played this game 8 times. And so here's a sample of some search strategies people had. I know, so it's like very like obviously the first thing you notice is some people are like really methodical about how they rate the environment, and like people are just like, I don't know, it's like just go somewhere, great. And so I've also ranked them from like from low exploration to high exploration, and I think you can clearly see what's happening there. You notice people who are kind of low on the scale, they tend to end their guess a little farther away. So I'm going to talk about that. So what I want to know is how accurate people are, right? So what I've done is I've taken the location of where that scent plume is. I can't point it out. So that red plume right there at the very end, I'm trying to figure out where that is in relation to how far away the person is, right? So the lower your score, the more accurate you are. Okay, so talk you through this graph. On the x-axis here, there's the conditions of the people, right? On the y-axis is your distance to the source. So again, you want to be low on the scale. That means you're doing really well. the lines are color-coded by how good the sense of direction was, 7 being really high, 2 being the lowest. Nobody was a 1, which is great. Nobody out there is bumbling around, walking corners. I think it's really nice. This is a really clear result that's just showing that the better sense of direction, the closer you end up to the odor source, right? But also, between the mono and stereo condition, there's a significant difference. So people who had the stereo inputs are doing a lot better than people who don't have that. It's not significant between stereo and flipped, but I think just by looking at it, you can see that there is some kind of change that's happening there. And another nice find we had was the cleanest result I'm ever going to see probably in my life is that between men and women, the better sense of direction is, the more accurate you are. And for people who know spatial cognition, this is like a pretty robust finding. Men tend to have, okay, great. So we determined that men do better than women in terms of spatial like sense of direction and how close they're getting to the odor source. And another thing that I was really interested in, of course, is this whole casting behavior thing, right? So I forgot to point out in this past screen here, but if you look, there's like gray lines, light gray ones and dark gray ones, right? So the light gray lines, you can think about as like exploratory behavior. People have not found the plume yet. The dark gray is when people are actually in there and that kind of casting behavior occurs. So an important question to ask is, does casting behavior actually do anything for people or animals? Like, does it actually help you get closer to the odor source? So what I did was I took this like in casting behavior section and I made it so I tracked which way people were going. And anytime anybody made a directional change of more than 45 degrees, I counted that as a single pass. Then, there's a nice little graph here, which shows that the higher the number of casts when you're looking for the odor source, the more accurate it's going to be. Another here, another graph here is really confusing, but all I want you to take away from this figure is that between two sniffs, if a person does not detect a big change in the concentration of odor, they're going to walk a lot farther before they sample again. And why is that, right? So we have to remember that animals and people are looking for the edges of scent cones and trying to follow that up to the odor source. So if you're standing in a plume and like you're at the max level of like it smells very strongly, but then you step like right out to the side of it, you're going to have probably like 0 or very low odor rate. In which case, if you're close, like just kind of step back to where you were. But in terms of like if you're in the middle of this area here, if you have a very low change in odor concentration, it means you're either not in the odor plume at all, or you're like really far down the gradient, and therefore you should just like keep going in one direction until you sample again and redetermine where you are. Okay, cool. So those are the first 2 questions I was hitting at, right? So can we, are humans actually casting? And also is stereo olfaction important? And so the third question I had, which was using people and animals as kind of like this odor detector proxy, right? What happens? What I did was I took all 179 people who played this game, and I just made a heat map of where they're walking inside the game and the down. Looks just like this. And so it might not mean much to you from looking at it now, but if I pop up this figure here, that down there, that's the mean odor concentration of a plume over time. And this is the same plume that we use for the game. And you can see we took that image and just kind of like rotated it around. It actually like really nicely falls upon where people are searching the most. So that's pretty cool, right? You can use people as an odor detector. Okay, cool. So we've shown in the game this seems to be like a pretty good system. So let's like take what we learned from the game and try to apply it to a real system of people walking in a real room. So disclaimer before I start with this is that I'm still crunching the numbers. I only have like a third of them done, so the results are coming in. But I'm going to show it to you anyway. So this is the person in the psych education library, which is now totally defunct. At the end of the room here, those white things, those are diffusers. And there's 12 diffusers, and only one of them is releasing scent of aniseed oil down the room. The rest are just dummies, right? And so we just asked people to identify which one of those 12 had the scent. And the reason there's a grid on the ground is because like you and I being smart people, if I told you like, hey, 12 of these things, like one of them has a scent coming out of it, telling you which one it is, like all of us would just walk to the front of the room, be like that one, great. So I actually want to start casting. So I forced people to stay back in what I call a zone for 30 seconds. They got to walk around or not if they wanted to. And then at the end of 30 seconds, they just had to make a guess for me. Like, which one do you think is in? And then move forward on after that. So right now I have 27 people coded, 37 people I have not coded yet. My wonderful RAs, Masha, Andrew, right, Sierra, they sat there like all summer and like winter and spring coding these videos for me. We got a lot more to go. But right now we're not seeing anything significant from the results. But if we just look at this like graph here, this is showing like for each zone what a person's average guess was. And I've also plotted if they have a right nostril blocked versus the left nostril blocked, and a stereo, which is just using your nose normally. And you can kind of see at like the very end, like stereo condition is like they're in the middle. In the right condition, they're like off to one side. Left condition, they're off the other side. So maybe given finishing the other 37 trials, we'll see something, but time will tell. What I have done is gone ahead and mapped the heat map of where people are going though, and you see a very similar structure to what's happening from the virtual environment experiment, which is that people seem to be sticking to where this plume is, and you kind of get an idea of what this plume looks like, right? Obviously it's very strong over here, people spend a lot of time in front of it. When they're back, they're kind of like spreading out and just like walking randomly. Well, the one cool thing that I did find was that in addition to tracking where people are going, we're also looking at what their sniffing is. They're wearing microphones around their necks to record the respiration rates. Again, hand labeling everything as of now. We're going to eventually try to do some like neural networks on this and try to like pull down machine learning. We'll work on it in the next like week or two, see what happens. But just by like hand labeling all of these, we've shown that people, when they get closer to the odor source, they sniff a lot more intensely. And what that does is when you sniff really hard, it actually separates the background odors out and kind of makes you like focus on that one. So there's something here to be said about how you sniff and the closer you get something when you're sniffing. So for the next steps though, what I'm trying to do with Lucia is when we go to the East Coast at Harvard, we have another collaborator, well, several collaborators over there. One of them works on olfactometers, which is just like a cannula basically, where you just put it to your nose, or in their case, they're using mice, and you just pump out odors in very specific concentrations to each nostril. And so we have a computer game, right? So we can track where people are moving. We give them an olfactometer, and hopefully we can actually now correlate like where you're standing, this is the odor concentration, we're gonna feed it into your nostrils, either stereo or mono, or we flip it, we see what happens. And like the most optimistic case, which we were really hoping for and I think is totally feasible, is you give people this game, give them the olfactometer, and then track what's happening inside their brains. I have no experience in doing this, so if anybody knows a postdoc that would love to do fMRI studies for a year, like, let us know. Okay, cool. So that's all great, right? But everybody here is like, whatever, humans, we came here for the dogs, right? Mark and Capo, I don't know if they're still showing up, but maybe they will. So in terms of like dog cognition, obviously my background is in animal cognition, right? So this study has been like super rewarding for me. We had this awesome ability to work with the California Rescue Dog Association, that's CARDA, and that is like the largest volunteer search and rescue team in the entire United States. These are people who come on their own time. They go after work 3 days a week, and they train their dogs and puppies to adulthood to just look for people. They like pay for all their own gas costs, everything. These are 5 people that we worked with here. Some of you might know Shay, if you work with animals. She's the one who actually emails you and is like, hey, your protocols are not up to date, or like, we're going to do a room walkthrough. So if you ever run into her, ask her about what she does. It's really cool. So working with CARDA has been super rewarding. And a lot of that is because despite like, I think our Wikipedia this is like 180 years of like certified search and rescue teams in the United States, nobody has really looked at search and rescue dogs and what they're actually doing in like a scientific framework. All these teams, they're training each other just almost like anecdotally, right? They just like bring on the rookies and they're like, we know this works. This is how you train your puppy. If you like train this way, you're going to be able to find a person. And they have like an 85-90% success rate. It's super high. And so it's curious why nobody has gone and like rigorously studied what these dogs are actually doing. And the handlers have such an intimate knowledge of what odors are doing in space and like where they're moving around, that we've learned so much just by hanging out with them, right? A lot of my hypotheses are actually like, people say something and I'm like, really, is that true? Like, can we just test it, right? So a lot of our work is actually just like kind of verifying what they already know. And then hopefully from what we've already showed them is true, you can change the way you study or train your dogs and improve your search efficiency in the field. So anyway, fun fact here is that there's actually a lot of types of search and rescue dogs. Stereotypically, we think of trailing dogs. Trailing dogs are the ones where you like take a shirt, you give it to a dog, the dog sniffs it, and then it like goes off and finds a person, right? I'll talk about the other types, but we're gonna focus on the trailing dogs right now. Okay, so weather effects and odor dispersion. What we did was we asked people to come and meet us 3 times of the day because we wanted to get things like the temperature, humidity, wind direction, wind speed. So we went out in the morning, the afternoon, and the evenings. This is in Brianna's Regional Park, just like 15 miles east of us. We had a half-mile long trail that we laid out. And when I say laid out, it means that we gave all our like little missing people a GPS, and we gave them a map, and we're like, follow this trail exactly, this yellow line right here, right? Follow it all the way to the end, and then just pop, squat in a chair we provided for you. We'll come find you in like an hour. So all these other like markers here, that's every 25 meters, we actually have a weather station and we're walking along behind people and we like stop and we record all those meteorological readings, barometric pressure, humidity, temperature, wind speed, right? We measure it when people go out, and then right before the dog goes out, or right after the dog goes out, we're also like hustling around behind them, measuring them again. And on the dogs, we had given them a GPS recorder and also a device that tracks their head position. The head position of like if they're trailing up or down on the ground. So what this looks like here, I'm showing you two maps. So this is an afternoon and a morning session. That black line, that is where the people are walking, right? And the black squares is where we have stopped and taken the meteorological readings. These gray lines are where the dogs are going. And the gray little circles, that's every single time this dog named Gig puts her head down on the ground. And the blue arrows, those are the vectors of wind speed and magnitude, or wind magnitude and direction, sorry. So the first thing to notice is afternoon, this is like a 90-degree day with like pretty low humidity. Morning was like pretty cool and pretty humid. You can already see some differences happening where the dog is, right? So we're looking good. Total trials I ran was 14. We have total weather station points though was 434. So I'm gonna talk about data in two ways. One is looking at the overall averages, the conditions over the trails, and then also taking like all 434 points and then seeing where the dog is in relation to those weather station readings. So in terms of like looking at overall average trail conditions, what I'm doing is I'm taking the area between where the person walked and where the dog walked. And the smaller that area, it means that a dog was sticking closer to the original person's trail, right? And if they're farther away, it just means that the dog is somewhere off in the distance. And I'd like to emphasize that it does not mean the dog is wrong when they are very far away from the human's trail. What it means is that the odor has gone over there and the dog is following it. And the dogs can be wrong. I'll show you like a little image of one where it looks like it kind of got lost, but most of the time they're still on the trail. They're just following it somewhere else. So what we found pretty robustly is that dogs stay closer to the original path when trailing if humidity is really high. And so there's a nice correlation here just showing if your humidity is down at 80%, then the area shrinks down. This is just confirming what like almost all the handlers intuitively know. They just know if they search in the evenings or in the mornings, the dogs tend to do a lot better job. If it's like really hot out, not only do the dogs not perform as well, but the dogs like also overheat, people overheat, right? You gotta take a break. The volatiles are up in the air and they're going somewhere else. And another thing that I thought was interesting we found here is dogs like work at different speeds. They're actually usually hauling pretty fast. Like this dog over here, these guys are going between like 5 meters per second to 8 meters per second. That's like a run. They're pretty much like dragging the handlers down this half-mile trail. What we've seen is that the faster a dog goes, the more often their head is actually up in the air. And then the slower they go, the more often their head is down on the ground. So that means dogs have two strategies, right? They can move really slowly and they can trail on the ground, or they can go really fast and use air sampling to find the person. Okay. So another way to look at data then is to look at every single weather station point here and just see how far away the dogs are from that weather station. And what I found is backing up the original claim from looking at overall trail conditions, that again, the higher the humidity, the better a dog is doing. And to clarify that temperature does have an effect. It just is that temperature and humidity are covariate in my data. The higher the temperature, the lower humidity, so I just like tossed it out. I was using a linear mix model, so I took like everything into consideration for this. I looked at like temperature, humidity, wind direction, wind speed. I looked at the dog, like the day they started, what number trail they were working on, the order the dog went in a day, because there was a huge like concern that dogs actually just follow each other all the time. So if you went first, you would do the first, but if you're the last person to go that day, your dog's following everybody else, there's no effect. The only one that came out was this relative humidity measure. In terms of like head down behavior, we kind of wanted to know those kind of meteorological readings that kind of define if the dogs have their heads up or down. Confusing finding, we can talk about it if somebody has some ideas. Again, relative humidity, the higher it is, the more often the dog is putting their head on the ground, but also like barometric pressure. So for people who don't know, the higher the barometric pressure, the clearer the weather tends to be. That tends to predict the dog is sitting on the ground or not. Like maybe you can speak more about this. That one paper we just recently found like 4 days ago, which is showing that like people tend to have a better odor threshold in terms of like high barometric pressure or slow, and then if it's more humid or not. So something is linked here, but there needs to be like a physiologist who goes and like actually studies mucous membranes and tells us like what the effect is of humid days and pressure on being able to smell. So another thing that's important here is, so this whole effective wind on navigation, right? I think intuitively we all know or think that wind should affect how well these dogs are trailing. Based on data I've collected, I haven't really seen any of that. So the higher the wind speed is, maybe you like kind of squint at it, it looks like perhaps the dogs like actually do a little better, they're closer to the trail, but I mean, that line I plotted is like going the complete opposite direction, right? Wind direction, you'd think a dog also would be blown off course or something like that. Again, it's like a mess. There's no correlation there at all. But there is like one graph that I saw that was interesting, which is this one. And what I'm showing here is on the x-axis, that's the wind direction. And I've just put in the east, northwest, south, east again, so you kind of know which way it's going, because it's like obviously a circle. And then distance to the trail on the y-axis. And there's something to be said about when the wind is blowing to the west, the dogs tend to be a little farther away from the trail. And when I saw this, I was like, I don't know. Like, I don't understand why this would occur at all. And I showed this graph to the handlers, and I'm immediately like, oh yeah, I know why. Like, they're just doing this so long, right? Turns out just like pure chance, when I was laying out this trail for people to walk on, this black one over here, we had people walking like almost straight east the entire time. And if the dogs were walking into a headwind, they tended to do worse, right? Their wind's blowing this way. And I think, at least for me, I always thought an animal would track better if it was working into the wind instead of blowing towards them, right? Well, the handlers explained to us that what actually happens is that the freshest scent is being blown from that person over there towards the dog. And you had to remind yourselves that animals work on the edges of scent cones, right? So if all that fresh scent is blowing towards the dog, it's actually stuck back here, and it has to like get out of the scent cone and then like walk back into it to figure out where it needs to go. So in that case, then a place this big distance from trail things are to happen. But instead, if you like flip it, right, the wind's blowing the other direction, the dog is very easily able to stay in the edge of the scent cone because the freshest scent is actually in front of it, it's on the trail, right? And I think this is like really amazing to think about, because it means that dogs are not only extremely attuned to which way the wind's direction is blowing and the odor scent cone, what it looks like, but also that just from like a 1 hour old trail and from odor particles, dogs know which direction a person's walking in, right? They can go in one direction and be like, oh, that odor's getting older, let me just turn around and go the other way. And I've seen dogs do this multiple times. It's kind of like the closest thing to magic, as you said, right? So this is a pretty cool finding. So, but this only really applies to trailing dogs, right? Because they actually care about the footsteps of a person. I'll talk about other dogs in a little bit, but other dogs tend to like, once they smell a person upwind of them, they'll kind of run in that direction willy-nilly until they find a person. Right. So we also are working with Ora Peleg, who also is now at Boulder. She's an awesome modeler. She's been studying dung beetles. So dung beetles, we don't know, they're like that little beetle that like stands, like does a headstand, like pushes a ball of elephant poo around the African savannas. And they're like pushing them towards like burrows, I think, to lay eggs in them or something. Apparently they like hop onto their ball of dung and like look at the stars and like orient or something. I don't know, it's crazy. But this is what Ora does. She models animal behavior. She used to be the postdoc, grad student in Mahadvan, postdoc. Yeah, so they're just really interested in like modeling cool animal phenomena, right? And so we talked to her about this, and she was super excited by the dog data. And she had this theory of like, oh, maybe dogs like do this dual like strategy of like sniffing really slow on the ground or fast in the air. And I'm going to show you just a quick model she made. So this model here, this is, imagine if a person's walking this half-mile long trail and there's like no wind in the environment and the odor's just diffusing. So it would look something like this, right? It's just kind of hanging and like kind of grading out. This is ignoring any like heavy particles that might stick on the ground. Just think of this like volatiles. Oops. And then she added the wind condition. So using some data from the dogs and what we had collected, she modeled what odors might do if there was a wind condition blowing it away, right? Pretty awesome animation. And what I've done is I've overlaid person's trail and then the dog's trail there, right? And remember how I said the dogs like very rarely get lost? You can see right here in this little section where the dog kind of like does a little circle. When dogs actually do lose a scent, they kind of like, I think almost all of them, they just either pick a direction, they just do a circle because they're looking for where they got lost, right? And you can see here, the wind in her model, it blows a scent downwards and the dog's no longer standing in it. So it was like, oh, what happened? And once it looped down, it found the edge of the scent cone. It was like, okay, boom, let's go. And then another thing to notice is this is a little switchback I purposely put into our trails because the handlers at some point were like, you can't switch back. Because you switch back, the dogs will know. They'll detect this odor over there and they'll ignore the entire trail and just like cut across the entire thing. And I was like, is that really true? Like, is that going to happen? So I was like, I'm going to put a switchback in this trail, right? And every dog, without fail, they cut across that switchback. And you can see, right, from Oric's model, you can see the scent like really like concentrates up there and like blows off in that discrete direction because they'll just ignore the fact that there are footprints in the ground. So it's interesting. And so after this, we've completed this like sort of neological effects on odor dispersion kind of experiment. We're now really interested in looking at what area dogs do. And so what an area dog is these dogs don't care about what the person smells like. They're trained to just like look for a living person. So think of like catastrophes like if you have an avalanche or a building goes down. The first thing to do is send out these dogs and they just are made to find a person, bark, and be like, hey, it's a living person. Tragically, if you wait a few days, there's actually specialized dogs called cadaver dogs, and they're made to look only for bodies. But we care about what odor these area dogs are doing. And so kind of like the experiment we did with the virtual odors, we just want to know you like flop a person down. In this case, there's a tree back here. I think it's like about a quarter of a mile. We just threw a person behind a tree and was like, hey, let's wait out here for a while. And we released dogs, and you can see these are two different dogs. They have this kind of like fasting behavior that happens, this one more so than the other one. And so we just want to do the same thing, measure all the weather conditions and see what affects where these dogs are going. And the other thing we really want to do is look at the temporal effects of odor dispersion. So this means how long can a dog actually search for a person once they've been missing? Because right now, Cardiff has this kind of like unwritten rule that if a person's been missing for 72 hours, they won't send a team out to go find them. They think the odor trail is too old. And so we want to actually like rigorously test like what is the cutoff, right? If you give a person the right conditions and the dog's the right conditions to look for a missing person, maybe if the whole time this person's been gone for like a whole week, it was cold, it was humid, and there's like no wind, it might actually be worth going out to look for them. But if it's been like dry, scorching hot summer day, maybe it's actually like 24 hours, like the chances that dog find a person have gone down dramatically. But there's also this flip thing too, where we think of like weather changes a lot. Even if the weather changes dramatically in that 1 week period, what if it just comes back down to being cold and humid again? Like, are there actually odors left in the ground that the dog can still follow to find the person? So this would be really cool, pretty rewarding to figure this one out too, right? It would drastically change the way people search for missing people. Okay, so concluding here, my first hypothesis was that mammals exhibit similar odor tracking behaviors, this casting behaviors, as all these other vertebrates and invertebrates I talked about. And I think we can pretty confidently say that yes, human and dogs are definitely exhibit casting behavior. And like I said previously, we have collaborators also studying mice and this behavior, and we're kind of seeing the exact same thing too as well. So it's been a cool study. The next thing is that we think that having a pair of sensory structures improves olfactory navigation. And I'm putting this with a yes with like a question mark because in my virtual odor experiment, we can see that there's definitely an effect. I think the pending data is the one with people in real life. And then the really cool question would be if we put people in the fMRI and they use an olfactometer, does the stereo effect still exist? But at the same time, these are paired structures that are conserved throughout evolution. So something has to be really important about them, and that alone gives good evidence that they're probably helping in navigation in some way. If you don't believe me, this bottlenose dolphin that I pointed out before only has one blowhole, right? This guy right next to it, that's the blowhole of a humpback whale, a baleen whale. And it turns out that a bottlenose dolphin is a toothed whale, and toothed whales all have echolocation, right? And it turns out their main olfactory system, the sense of smell in their brains, is like highly reduced. They just don't smell anything really anymore. And remember, like all cetaceans, those are like animals that came from land and moved back into the water, right? And evolution doesn't think it's a useful structure, it gets rid of it. But in other terms, these baleen whales, they eat krill or like small fish. And there's one paper, I think, Risha, maybe more than that, that just shows that when humpbacks or something else, like they come up to the air and they like take a breath, they can actually like, smell krill on the winds and they'll orient towards it. So in that case, they're still using their sense of smell a little bit, right? So in that case, keep your two nostrils, something about it is really important. And then we also have another collaborator who's modeling this. It's our final collaborator I'm going to talk about. They're mathematicians, and I don't even understand exactly what they're doing. They're using the Shannon uncertainty principle. Maybe Carlos, you know something about this, we can ask you about it. What they're basically showing is that information saturates very quickly if you only have one structure sensing the plume. So what they've done is they've taken John Camolli's data sets of the plume, and they're just asking like, where is this person inside of a plume, right? If you have one structure, the information saturates quickly, you're cut off. If you sample quickly, then you have a little more information. But if you have two sensory structures and you're sampling relatively quickly, you can actually figure out where you are better inside of that scent plume. And then our final question is this whole idea of like using animals and people as a way to study odor dispersion in the field. We are confidently able to say that yes, dogs are able to track odors outside, and they can give us an idea of where odors are dispersing. Most likely, any other animal out there will also give you a pretty good idea of where odors are in the field. Very confidently, I would say that when you're trailing an odor outside in cold, humid conditions, the odor is not dispersing very much. And we don't really know why. We were looking at papers for this a couple weeks ago, and there's apparently papers in pesticide literature, and the theory goes that there's a lot of bonding sites and substrates that are competing for the odor, right? And so instead of being able to bond, they kind of bounce off because the air is humid. Maybe just hang around on the ground. That's why dogs put their head down a lot. Odor isn't going very far.